As Sylvia mentioned, I'm the business development manager responsible for Europe at Alvéole, and I would like to briefly explain you today our flagship product, the PRIMO bioengineering technology. I will give you a brief overview about our company first, then go through a little bit about the workflow or how we can achieve your custom microenvironments, and show you just briefly a few examples about classical micropatterning experiments, and then I will leave it to my colleague Aurélien, who will then continue and deep dive into 3D bioengineering using the PRIMO technology. So first, a few words about Alvéole and our team. We are a French company based in Paris, and it's actually the company was founded in 2010 by 3 researchers which have expertise in physics, neuroscience, engineering, and microfluidics. And they had this idea to generate a device which can actually enable you to generate your own bioengineering tools in your own lab for cell biology. And this is how we started off in 2010. Now we have actually reached roughly 15 people. Some pictures are missing here, but the good news is that the company is still growing. We have currently 4 offices, 2 in France, 1 in Germany, and 1 in US. And we already received 2 prizes for the flagship technology PRIMO in 2014 at the World Innovation Challenge, and in 2016 for the New Product Award at SLAS. Just a brief introduction about our ambition at Alvéole. Of course, PRIMO is not our only product, but it reflects quite well our vision. We want to generate tools at Alvéole to engineer and tune the microenvironment of cells, and our production, our R&D, is working towards developing these new tools. The logic behind this is quite simple. You have probably all worked with in vitro and in vivo systems, and you may know that usually in vitro systems are actually rather easy to use. Unfortunately, most of the time they are not highly physiologically relevant. And vice versa, in vivo systems are usually more relevant, physiologically speaking, but of course, since these are living organisms, they are not so easy to handle, and we also want to reduce animal testing. So this is why at Alvéole our vision is to develop these engineered microenvironments where the scientists can decide their best conditions by controlling chemistry, topography, and rheology of their own cell culture to make in vitro systems more physiologically relevant and reflect the in vivo situation more. So how can we actually control these microenvironments better? For this aim, we have developed the PRIMO bioengineering technique. So it's actually a contactless and maskless tool which needs to be connected to an optical device, an inverted microscope in this case. And the system is composed of 3 components, 3 major components. The first one is the optical box, the second one is the Leonardo software, and the third one is the photo initiator PLPP. If you would open this device, you can see a schematic overview of what is actually inside this optical box. You can see that we have a UV laser at 375 nanometers, a set of mirrors which will guide the UV laser in the end to a micro mirror device, a DMD. And from the DMD, the light will be reflected and guided through the optical light path of the microscope. So the illumination pattern is strictly controlled with our Leonardo software, and the reaction which will enable to pattern biomolecules is controlled with the PLPP photo initiator. So how does it actually work? First thing that you have to do with the PRIMO tool is to design your pattern, and this is where we are already quite different compared to existing techniques. So you are actually completely flexible in designing any size, any shape of pattern using your own desired tools. So this can be as simple as paint, or you can also use more advanced drawing softwares such as Illustrator, for example. The only thing that you need to keep in mind is that in the end, you will need to have a grayscale image and a TIFF or PDF or PNG file format, which will be then recognized by the Leonardo software. So the bright or gray areas in the design that you achieve will be projected in the exact shape and size through the optical light path of the microscope with UV light. And then this UV illumination can be used for mainly two applications. I will briefly talk about the functionalization of the surface. So to say, the micropatterning of biomolecules on the surface. And my colleague Aurélien will mainly talk about the substrate structuration, which can actually be a microfabrication of 3D microenvironments or hydrogel structuration. In addition, you can also combine these two approaches and first generate a structure in 3D and then functionalize it. And Aurélien will also talk about this approach later. So first, how does actually micropatterning or patterning of biomolecules work on a standard cell culture substrate? If you would go through the workflow, let's say you're starting with any type of substrate that you would usually use in your cell culture experiments. Let's take a glass cover slip or petri dish. The first step that you need to achieve is always to passivate your substrate first. And this can be done by coating your substrate in solution with an antifoiling coating. Usually these coatings are PEG-based and they prevent the attachment of any biomolecules or cells onto the surface. So once you have made sure that your surface is passive and nothing can attach on your surface, now the real important steps for micropatterning start using the PRIMO technology. So the next step after passivation of the surface is the addition of the photoinitiator, the PLPP reagent in solution onto your passivated substrate. Then you may choose your desired image, load it to the Leonardo software, and this image will be illuminated in UV light through the optical light path of the microscope onto your sample. I mentioned before that the UV light here is at 375 nanometers, and in combination with the PLPP reagent, the illuminated areas are activated. So what does it actually mean? The photoinitiator PLPP is activated by UV illumination and starts to locally degrade these antifoiling PEG brushes that you have just applied in the second step onto your substrate, making these sites of illumination accessible for biomolecules. These can be standard ECM proteins or also some other biomolecules. They attach to the surface, and then after a few washing steps to get rid of the excess proteins, you may now add your cells onto the substrate, and the cells will only be able to attach to those areas that were illuminated and that are now accessible for proteins on the surface. I will give you a few examples about the performance of the system and then show you further some examples about how our customers are actually using this technique in their own research. So first, I think it is quite important to mention that with this kind of contactless and maskless technique, it's really easy to align and attach multiple proteins on the same substrate. So on the left panel, you see an example where we actually patterned two different proteins in red and in green onto existing 3D PDMS pillars. And since we use a digital system to align, we have actually a quite high precision. This is below 1 micron, and you can precisely locate your proteins on your desired sample. Second, you can actually control the illumination and the dose of the UV by controlling the flipping rate of the micromirror devices. To put it very simple, by controlling the UV dose, you can control the amount of proteins that will later attach to the surface. So let's say you project a grayscale image which has bright spots and also dim spots. like this gradient that you see. In the end, this results in differential attachment of proteins on your sample, and hence you can control precisely the control of biomolecules on your substrate. This can all be done with a relatively high subcellular resolution of 1.5 micrometers. This is just one example of dots that were patterned next to each other in the field of view. And the good news for all cell biologists among us is that we are compatible with multiple substrates. I mean, anything that you can basically think of using in your cell culture experiments can be used in combination with PRIMO. Let's say you are interested in using slides, cover slips, petri dishes, even 3D devices, microfluidic devices, and PDMS layers. They will be compatible with our PRIMO technology. And one additional slide, maybe to clarify a little bit more how this quantitative approach works, because this is quite important also for the microfabrication step later on. You can, in principle, control this by designing this digital image file in the beginning and having different levels of gray on your file. And basically what happens when you project or when you upload this image to your software is that the areas that are bright will be illuminated throughout the whole experiment, and the areas that are dim will be illuminated less according to their grayscale level throughout the experiment of let's say, illumination. Which means that when you illuminate more, you degrade more of this passivative layer, the PEG layer, resulting in more attachment of biomolecules on the substrate. And if you illuminate less, then you basically have less space amenable for proteins or biomolecules on the substrate, hence less molecules attached. And this can be nicely controlled, as you can see on the right, with the UV dose that you apply. So if you increase the UV dose, you will increase the density of molecules as well that can attach to the surface in the end. Now I will show you briefly just a few hand-picked examples to show you, to describe how our customers are actually currently using the system. And every day we actually get new ideas from them, and it is a quite flexible system, so this should not be restrictive. But mainly, if we would look at the applications from our customers, we see that some of them are using this to pattern single cells on various substrates. You can see on the left panel, this is an image provided by Manuel Terry's lab, that they use the system really to study the cell cytoskeleton, and they force the cells into certain shapes to see how this affects the focal adhesion formation in these cells. In the middle, it's just to show you that this technique can also be combined with other techniques. So this is a collaboration with Alvéole, and here we show that these patterned cells can then nicely be imaged in super resolution in 2D storm using the system of Alvéole, but also other systems. On the right top panel, we patterned some molecules on some cells on ligands. So you can see the cells here, the green transmembrane protein that is accumulated on the patterned ligands. And this way you can actually study how the cells behave on different types of ligands. And of course, if you would like to work on populations of cells and not on single cells, you can do this just by changing the initial image that you design. So you can design it such that you have maybe cell duplets. In this case, you're seeing 2 human fibroblasts, or you can also study larger cell populations, such as in this case, where we just patterned our company logo basically on fibronectin micropatterns. You are also not restricted with standard ECM molecules. Again, on the left side from Manuel Terry's lab, you can see that you can also generate in vitro assays using this technique. Here they have patterned ARP2/3 activators, and in the video you can see the actin polymerization on these patterns. On the right, we have shown that you can also pattern antibodies. And you can also pattern antibodies or other molecules in high precision next to each other. As you can see, the alignment of these patterning experiments is quite efficient, and this will provide you a lot of flexibility to attach multiple molecules next to each other. One of the, let's say, hot applications that came out a couple of years earlier is to pattern actually more exotic substrates such as electron microscopy grids. And I'm giving this example because this is actually solving a major issue in this field. The problem is that when you usually seed your cells on electron microscopy grids, they would attach all over the grid, and you would actually not be able to analyze most of the cells, because to be able to analyze them, they need to be centered and they need to be in the middle. So there is need of a cell confinement method, and this is where PRIMO comes in. Since we are a contactless technique, we can actually work with these kind of fragile substrates. And on the left side, you can actually see that we have the lab of Kai Grünewald has contributed this to this experiments, and they have successfully patterned with GFP some circular micropatterns on the carbon layer, and on the right you can see that the cells actually respect this micropatterning and they attach to the middle. So in the end, they have high yield of cells amenable for cryo-ET imaging. If you would like to go and do these experiments in high content screening mode, this is also possible. So on the left, you see large areas that were patterned with the PRIMO technique. And this is especially useful if you would like to, for example, test the effect of different drugs and how these drugs affect the cell morphology or specific compartments within the cell. And on the right, you can actually see that with very high efficiency, the cells actually can attach on these green fibrinogen patterns. And you can study multiple conditions in a very small dish. Next, this is an example where our customers, they patterned cardiomyocytes, and this is quite interesting because cardiomyocytes maturation and proper function in vitro is highly dependent on their shape and microenvironment. So this is actually one of the specific cases where micropatterning is inducing maturation of a specific type of cells. In this case, you can see that these cardiomyocytes, they behave differently when you pattern them in populations, in duplets, and in different shapes. So especially the sarcomere organization in red, and also the gap junctions that are formed are different in different types of patterns, which is actually enabling the researchers to test multiple conditions and to find the best conditions that will give the readout about the most mature cardiomyocytes. And then these actually can be further tested by performing additional assays such as physiological readouts of cell contraction. In this slide, I would like to emphasize that since we can actually pattern biomolecules in different concentrations, this can easily be used to study, for example, cell migration. In this case, this is from Aurélien Rous's lab in Geneva, and they have actually patterned the same molecule but in different concentrations in this case, and it's a mix of fibronectin fibrinogen. And we will see now when I play the video how the concentration on the substrate affects the cell migrative behavior. As you can see on the left panel, the cells tend to stay on the highly concentrated area here. On the right, it's a similar experiment. In this case, still the same fibronectin pattern, but in the end of this road, we have a gradient in this case, and again showing that you can actually control nicely the quantity of proteins patterned on the substrate. Here you see that the cells kind of go a little bit more towards direction, and they can sense the biomolecules on the substrate, but they again prefer to go on the higher concentrated areas on the left side. So you can imagine multiple assays like this to really test different biomolecules and how this really affects in vitro your migratory behavior of your cells. And this is the final slide. In this one, I would like to emphasize that since this is a contactless technique, you are actually quite flexible to work on also very soft substrates. You will not break your substrate while you are patterning proteins. In this case, our users here have patterned airway smooth muscle cells, and they are checking if the stiffness of the substrate has an effect on the maturation of cell-cell junctions. And they can see actually that when stiffness is increasing, the type of junctions that are forming in between the cells are different. And they also nicely combine this assay with force measurement assays, where they, prior to the patterning experiment, embed beads onto the gel, then pattern their cells. And by following the displacement of the beads, they can actually measure forces, and they can really investigate how these cells behave on different substrates which have different stiffness. So with this, I would like to finish this introductory part and move or leave it to Sylvia maybe, and then she will probably give it up again to Aurélien to discuss a bit more about 3D microenvironments and how you can actually design and build them using PRIMO.
I wanted to thank you for your very, very nice talk. And now I would like to just summarize in 1 minute in Spanish just the main point of your presentation. Pues en esta primera charla hemos podido ver que PRIMO es una tecnología de bioingeniería, trabaja sin máscara y sin contacto con el sustrato y que permite, entre otras aplicaciones, crear micropatrones con biomoléculas para prácticamente cualquier cultivo celular, y permite que los usuarios puedan controlar todas las señales bioquímicas en los experimentos en cultivos. Trabaja con un fotoiniciador que se llama PLPP, que está patentado, y con un software llamado Leonardo, y con él se puede hacer el diseño de cualquier patrón, y también es posible controlar la cantidad de biomoléculas que se van a adherir al sustrato. Tiene una aplicación directa en ensayos in vitro y también en estudios de adhesión celular, migración celular, confinamiento celular. Es un equipo muy versátil. Carlos.
Pois, eu creio, eu acho que a apresentação em inglês foi suficientemente esclarecedora. Acho que não vale a pena estar aqui a fazer a tradução de inglês para português, mas já me dirão. Silvia, creo que puedes seguir.
Ay, perdona. A continuación me gustaría presentarlos a Aurélien Pasturel. Él trabaja como ingeniero en la parte de desarrollo y nos va a presentar una aplicación muy bonita con un trabajo en hidrogeles. Aurélien.
Yes.
We are looking forward to hear you.
Yeah. I was just waiting for the go. And so, do you all see my screen?
Now it's here.
Okay. So, good morning, everyone, and thank you for joining. So, as said before, I am Aurélien Pasturel. I work as an R&D engineer and mostly on hydrogel. However, today I will talk about a broader topic, which is using PRIMO not as a functionalization tool, but as a structuration tool, not only for hydrogel, but also for microfluidic devices. So as an introduction, when you look at the literature, you'll start to find that there are more and more microfluidic designs that are successful in biology, such as this chamber here with microchannels, which is very efficient for cell migration or axon guidance, and the hydrogel chip, which some of you may know because it helps you seed cells within an hydrogel in this compartment in the center, and you still have some room for cell-cell contact. So it's great for cell culture and co-culture. However, when it comes to microfabrication and structuration, there are many issues that you may come up with. First off, the optimization of the design is not always easy, especially if you have bought the chip or if you want to do it yourself. And if you want to go even beyond the original device and add some in situ modification with proteins or create some topographical features or multiple layers, you will end up with the many issues of alignment and actually reaching these confined areas. And I think this is where PRIMO really shines as a tool for microfabrication. So to prove that, I'd like to go back to the basics, which is what makes the core of PRIMO technology. At the core of PRIMO technology lies a digital micromirror device, the same kind of device you would find in a projector. It lets you turn a UV laser into a shaped beam with any combination of shape and gray levels you want. And there are three characteristics that I think make PRIMO good for microfabrication, which is first, the wavelength. The wavelength of 375 nanometer is great if you want to cure the classical resins, such as SU8, or if you want to trigger classical photoinitiators, such as Irgacure, LAP, Darocure. And on top of these chemistries that work with the PRIMO system, we have the access, as Mehmet said, to grayscale projection. And this grayscale projection, you can see it as an added layer of control you can have over your structuration, because many of the chemistry that you may use are actually sensitive to the UV dose or the UV power or both. And last but not least, there is the collimation. If you plan on working with high aspect ratio designs or making stiff samples, the fact that the beam is very collimated, which means that the shape is preserved along bigger thickness, is actually very instrumental. So it is no surprise that we are seeing more and more researchers doing structuration with PRIMO, either by directly building their microfluidic device, which will be the first part of this presentation, or by buying devices or making their own device with their methods, and then functionalizing and building inside the device. And this will be the second part. So without further ado, let's start with the first aspect, which is building the device. And one of the most obvious applications is replacing the photomask in soft lithography. Traditionally, when you want to build a microfluidic device such as this one, you have to start with a photomask to cure a resin. And this photomask can be long to obtain. Either you have the material to create this photomask, and it can still be a bit long, or it can be even harder if you do not have access to the material. And in such case, you have to order the photomask, which means that from the idea you have your design to the results that you want, there are actually many methods and many times where the thing could go horrid. So what we propose with the PRIMO system is that PRIMO can be your photomask. As Mehmet said, the Leonardo software can digest many kinds of different files, such as PDF files or big image files. And this centimeter scale image will be automatically dissected into separated field of view by the Leonardo software. It will align them automatically and also allow for some stitching in the red areas that you see here. So the stitching function has been optimized for the SU8 resin to make sure that you cannot distinguish between several fields of view. As you can see here, the Alvéole logo was made by 4 separated fields of view. And as you see on the picture here, you cannot see the mark of the stitching, which means that in the end, your product, the PDMS chip, will have no leaks. So one of our pioneering users that made use of this fast prototyping of a microfluidic device with PRIMO were the group of Manuel Terry at the Cytomorpho lab. They wanted to recreate the hematopoietic stem cell niche, which means a co-culture of no more than 5, even more different cell types that you have here, the UVEC, the HSC, MSC, and osteoblast. And so a simple chip like this one would simply not be sufficient for such a complicated co-culture. So over the course of their experiments, you can see that they almost went crazy with the design and ended up with this very complex microfluidic chip. Complex, yes, but it was still working, as you can see on the picture here. The chip was functional, and they successfully achieved the culture of their 4 different cell types for the hematopoietic stem cell niche. So I really like this example because it shows you how with PRIMO you can directly print onto the classical resins such as SU8. But there are more exotic resins which are also very interesting to test with the PRIMO system. We have a collaboration with the Institut Pierre-Gilles de Gennes in Paris and the Institut Curie on a positive resin that is sensitive to the UV dose, which means that when you combine it with the PRIMO system, which can control the UV dose with the gray levels, you can project gray level image and obtain a resin that has been topographically printed. So you have these images here, which are optical profilometry images. But I think it's even clearer if you look at the SEM reconstruction here. So you can see these beautiful ramps and those cuvettes here as well. So if you think about application in lab on chips, such as micro lenses arrays, ramps for cells, or the herringbone mixers, all these structures are actually difficult to create in the classical soft lithography process. Here with the PRIMO system, it's kind of easier because it's simply a matter of designing a grayscale pattern and projecting it onto your resin. So this concludes the first part. I hope that now you can see how you can use PRIMO to directly work onto resins to create microfluidic device. But some of you may have already their device in-house or have built their device. And so on the second part, I will show you that some of our users are actually using the PRIMO system not to build the device, but to build inside the device, to add features into an already complex system. So let's start with the most obvious application, which is using PRIMO for protein patterning, just like Mehmet explained, but this time within the confined environment of a microfluidic chip. PRIMO is contactless, so it's great if you cannot reach the channel, because as soon as the light can reach it, you can do the functionalization. So this work can be found in a review in 2020 from the team of Katarina Goss, where they patterned 2 different proteins to study the migration of a T cell along this thin channel. So the T cell could migrate all along the fibronectin micropattern on the first part of the chip. And once it reached an anti-CD3 pattern, the cell started to stop. So on this image here, which is basically acts like a chymograph, you can in fact follow the nucleus of the cell, which was tagged in blue. And you clearly see that when it reaches the anti-CD3 pattern, the cells start to stop. So overall, I think this is a great example of using surface functionalization to make your microfluidic chip a bit smarter. But what I really like also, and this is one of my preferred applications, I have to admit, it's using PRIMO to create structures inside microfluidic devices. So in this example, we assembled a very simplistic chip made of 2 layers of PDMS, and we filled the chip with a solution of 4-ampeg acrylate. 4-ampeg acrylate, it's an hydrogel precursor that can be photopolymerized with a photoinitiator under the illumination of PRIMO. I will show you in this example on the video, I hope it runs smoothly, that by printing several field of view, you can start to create channels inside the simple microfluidic device that was in the beginning. So now we have 3 separate chambers with each other, and we have some complex features. And just as a proof of concept, we incubated a solution of beads to show you that the channel is actually functional and can support a very simple capillary flow. And you see the beads are not crossing over the gel, so the channels are actually leak tight and functional. And I think there is really plenty of application with creating hydrogel channel, especially if you think about the fact that these hydrogel channels are actually not impermeable, they are semi-permeable. On this video, you can see that the small beds that are a few micron in diameter are stopped, but the water actually passes through the barrier made of hydrogel. So this was done previously in 2018 by the team of Jean-Baptiste Salmon in the LOF, where they used in situ photocross-linking of hydrogel to create this filtration barrier. And for the many biologists around, I think this may be a great tool, because you could imagine cultivating cells and having them communicate through their factors without actually getting in contact with each other. I really think there are plenty of applications with that. So overall, and I mean, even though this is very promising and very efficient, I have a small technical slide to tell you about oxygen inhibition. Many of you will be using PDMS-based microfluidic devices, which are permeable to oxygen. And oxygen is well known to inhibit the gelation, which means that if you want to polymerize your gel under our atmosphere, chances are that your gel will not polymerize up to the top of the PDMS, which means that you will not create channels. Those channels will be leaky. There are ways around this, however. You can first use other materials than PDMS, or you can temporarily remove the oxygen by putting the device that you have into a sealed chamber and perfusing argon or azote on top of it, and then printing your gel. So this was a quick slide just to tell you that when you want to do in situ photocross-linking, you have to take oxygen into account. There are ways to get rid of oxygen, and interestingly, there are also ways to use oxygen inhibition to your advantage. This is also an original work that we have done a few years ago, where what we saw was that the gel thickness can be controlled thanks to grayscale projection and oxygen inhibition. To explain what happens is that when you project darker gray levels, you have more oxygen inhibition, and in the end, your gel will be thinner. If you project brighter gray levels, you will have a lower oxygen inhibition and your gel will be thicker. So, as a schematic, if you project a grayscale, you will obtain in the end an hydrogel topography, just like with grayscale resins, but this time it is made of hydrogel. It's more than 90% of water. And the cells actually like it. You can do plenty of interesting things with those hydrogel templates, such as using PEG templates for spheroids assembly. So the PEG templates that are topography here are non-adhesive, which means that the cells actually cannot adhere to the gel, and instead they will adhere with each other. So you can use these PEG templates to create these non-adherent spheroid assembly templates where the spheroids are actually very standardized. But you can do more with PEG. PEG does not have to be always non-adhesive. You can revert that. And this is the last slide before I conclude, so I hope I'm not too long in time. To revert the non-adhesive properties of PEG, we have a protocol which is called decoration. To do decoration, we use a linker which is called acryl PEG SVA. We graft the linker onto the gel with the PRIMO system, and then the linker let us graft some biomolecules such as polylysine and fibronectin or lamellin. So that, as you can see in this example, these are neurons which are seated on the same PEG hydrogel as before, but this time instead of forming spheroids, they are actually spreading, and you can see their axons, etc. And very quickly, as a last example, you can go even further than that. You can combine structuration and decoration, which means that you can combine the creation of shapes with specific patterns. As you can see, the COS7 cells are actually adhering into the red decorated area and are also spreading all along the topographies. So you can make very complex cell culture templates thanks to the PRIMO system. So as much as I'd like to talk for hours about hydrogel, I think it is time for me to conclude this presentation. And so I hope that now you have a clearer view about what are the possibilities of PRIMO in terms of structuration, especially in the context of microfluidic. You can use PRIMO to make your device by directly printing resins, and you can use PRIMO to make your device smarter by printing inside protein micropatterns or hydrogel structure. So with that, I'd like to thank you all for your attention. Thank the member of Izasa for being nice all along this preparation and for this opportunity. And I am available if you have any questions.